astronauts aboard the ISS International Space Station have identified microbes in space for the first time without having to take samples back to Earth for testing. The ability to identify microbes in space could help diagnose and treat astronaut ailments under real conditions. Time, as well as help in the identification of life based on DNA on other planets. It could also benefit other experiments aboard the orbital laboratory. Identifying microbes involves isolating the DNA from the samples and then amplifying or making many copies of the DNA, which can then be sequenced or identified. The investigation was divided into two parts: the collection of microbial samples and the amplification by polymerase chain reaction (PCR), then sequence and identification of the microbes. NASA astronaut Peggy Whitson conducted the experiment aboard the orbital laboratory, with the NASA microbiologist and her team, who observe and guide her from the United States. As part of the regular microbial monitoring, the Petri dishes were touched on several surfaces of the space station. Working within the microgravity science glove box MSG about a week later, Whitson transferred cells from growing bacterial colonies to those plates in miniature test tubes, something that had never been done before in space. Once the cells were successfully collected, it was time to isolate the DNA and prepare it for sequencing, which allows the identification of unknown organisms, another first for the spatial microbiology.